Assalamualaikum and hello everyone. In this video, our group will be discussing the article Mini RNA Molecules Shield Mouse Liver for Hepatitis. Before we dive further into this article, let me clarify some of the terms. RNA is a polymeric nucleic acid molecule essential in various biological roles such as coding, decoding, regulation, and expression of genes. Next, according to the Centers for Disease Control and Prevention (CDC), hepatitis means inflammation of the liver to the point that its function is affected, which is often caused by a virus. So how can RNA molecules intercept the disease from spreading in a mouse liver? Will the same method be applied to humans with hepatitis? And most importantly, is this method safe to be applied to the human body? To answer this question, I will pass to our first group member, Fadhlullah.
Thank you, Izzati, for a well-explained introduction. Now, I will continue our presentation. When we learn biochemistry, we understand the importance of RNA, especially on synthesizing protein following the central dogma of life. But did you know that there is a study carried out where millions of RNA molecules are injected into the mouse's bloodstream? Guess what the outcomes? The mouse's livers are protected from the ravages of hepatitis virus. This technique used on the mouse is known as RNA interference or RNAi, where they utilize a short bit of RNA strand to silence a specific genes. In this case, they blunt the self-destructive inflammatory response that could be triggered by the agents like hepatitis B or hepatitis C viruses. This truncated RNA could turn off the very genes that generate them. The biologists are optimistic on this newly found versatility and potential that RNAi could offer. That's all from me. Now let's see what Arrshath got for us.
Let me continue. Judy Lieberman and Premlata Shankar was the first immunologist from Harvard University to carry out RNA interference against disease in an animal. Solution of a small interfering RNA, as known as siRNA, was administered into a mouse tail in a massive amount in order to protect it against hepatitis. Surprisingly, it reduced the inflammation of the liver and there's no any side effects found on those mice. As a discussion, RNA interference offers a great clinical potential as therapeutic modality, especially for inflammatory conditions. The delivery method of siRNA was hydrodynamic intravenous tailwheel injection. The mice were sacrificed over a time course. Representative tissue samples in liver were collected and analyzed by using fluorescent microscopy to determine siRNA uptake. That's all for me. Thank you.
Thanks Arrshath for the lovely explanation. Scientists can still induce autoimmune hepatitis despite of no presence of viral analog in animals. Because of the liver readily absorbs siRNAs into the cells, the disease became so popular in RNAi studies. Lieberman's team injected mice with siRNAs in a series of experiments designed to shut down a gene called Fas. When it is overactivated, during an inflammatory response, it induces liver cells to self-destruct. The next day, the animals were given an antibody that sends Fas into hyperdrive. All control mice were dead except for 82% of siRNA-treated mice. Between 80 to 90% of the liver cells had incorporated the siRNAs. Besides, the RNA molecules function for 10 days before fading completely after 3 weeks, 3 times longer than in previous studies. Next, I believe Halimatul have something in mind to share.
Thank you, Izzat. Now let's further move to the application of siRNA. Researchers had performed several tests, which include injections of concanavin A or Con A on several animals. Con A is a fun lectin which induces liver inflammation due to the activation of T lymphocytes. Animals that were infused with siRNA were not affected. Based on several lab procedures conducted in petri dishes, siRNA were found to be an inhibitor for the replication of hepatitis C. In addition, a researcher named Premlata Shankar and other scientists had discovered RNAi's ability to potently suppress HIV's infection, as siRNA can target CCR5, which is a surface receptor used by HIV to enter T cells. Apparently, the siRNA prevents HIV from entering most T cells and keeps it from replicating when it manages to slip inside. I'll hand over the next part to Izzati. Thank you.
In conclusion, we can deduce that RNA molecules indeed assist in preventing hepatitis from the mouse liver. Although the method can't be applied to humans, with enough research and innovation in technology, surely we will be able to take a step further into a new era of biomedical. That's all from us. Thank you.